Hi everyone. So today is a very short talk, so I'll be happy to take more details this afternoon in the poster session or if you see me anytime during the break. Anyway, markers. So a few of you have asked me what do I mean by that. So here as a marker, I define a genomic region that can be used to track pathogenic outbreaks. So in this case, a marker is a region that is unique to your taxon of interest, your organism or a pathogen, and is absent from all others. And you can use this to develop, for example, PCR primers. And ideally, this PCR primer should be highly specific and sensitive, so you are always able to pick your organism and you're 100% sure of what you're picking. Now, current approaches to do this compare your target organisms to their closest relatives, for example, their neighbors, to try and find out these markers. Now, one such program that does this is FUR, which was initially developed in our group, and uses this exact matching suffix arrays to find these target regions. Now, we had some tests in the labs, and this worked very well, but there were two problems, and I'm going to go through this and two solutions that we found of it. So even though the program was very useful, it actually had a high memory problem, so it could only be used to analyze small data sets because the suffix array was constructed on the entire of the neighborhood, so it would increase linearly as your data set would increase. So our solution was to implement a new algorithm which actually constructs a new neighbor so that the memory is now proportional to the size of your longest sequence, regardless of the number of genomes you're actually looking at. And now, another that is a bit more problematic, and we've been hearing it over, I think, these past 2 days, is that users needed to manually define what is a target, what is a neighbor, and it's crucial that it is actually very well defined, otherwise you may run into further errors downstream in your analysis. So in summary, this is basically my short talk is this in workflow for finding these marker regions, starting all the way from your string of your organism all the way to finding these primers. So in this three-step workflow, the first thing we need to do is define our targets and neighbors. So you start with your string of your favorite taxon. And what we want to do is to first define taxonomical targets, so which are all genome accessions that share the same most common recent successor or from the taxon ID. And then we define all taxonomical neighbors as those that share, if you climb one taxonomical rank above, those are all the taxonomical neighbors. And you can get this very nice list, but there is a problem which some of you may be thinking of, is what if the taxonomy is not correct? So that's why we made a second step where we actually infer from all of the genomes we downloaded, we infer a new phylogeny, and the aim is to detect a monophyletic clade. And this clade may or may not contain misclassified genomes. So you see a tree for Legionella pneumophila subspecies, and you can see that in the pneumophila clade, you have 6 red ends, and those are accessions that were initially misclassified in the database, even though they are phylogenetically closer to all other targets. So now that you have your monophyletic clade, you can now go on and find your markers. This is the second step with the program I initially mentioned above that now has this iterative suffix array construction. Basically what it does is it uses match-link to find homologous regions between the targets and the neighbors, and it subtracts them. And in the second step, it ensures that all your potential marker regions are actually present across all of your targets. And finally, there's a second subtraction with BLAST to make sure that you actually remove all cross-reactivity that may happen. And as I mentioned, the difference between the previous and the new algorithm, and you can see the new algorithm with the filled circles, is that the memory is constant in the size of the longest neighbor. So this is a neighbor simulated sequence, and they're all the same length. And you can see that even as you increase the size of the database, your memory keeps constant. So if you index your longest sequence on a laptop, you should be able to run your entire data set on your laptop, for example, as long as you're willing to wait for it, but it is also relatively fast. So now you have your markers, which are present across all of your targets, and if you want to do PCR primers, you want to convert these markers into a primer. So we use primer tree, and we select the lowest penalty score it can actually construct, And we score this with sensitivity and specificity by doing an in silico testing in the NT database. So how often does your primer match a session that it should, and how often does it have a mismatch that it shouldn't? And so, and this pipeline, we evaluated it at a relatively large scale, hence the title. So we grabbed this list of prokaryote references of 120 prokaryote references, and we went through all of them in this pipeline to try and find how many markers can we find, what is this sensitivity, specificity, and so on. So so right from the start, we removed 10 of them because of the low quality genomes. So we actually require minimum 1 target, 1 neighbor. For this analysis, we asked them to be at complete level. You can actually go make chromosome, but that's so far the minimum requirement. So out of these 110 prokaryotes, which also includes E. coli, so it's 2,000 genome sessions, something like that. We were able to actually find primers for, it's cut off, for 82 of them, and this is actually quite good. And when we actually evaluate this in silico, on average, they had a sensitivity of 0.98 and a specificity of 0.93. So this is almost basically perfect scores. And some of these primers had been previously tested in wet lab with our previous program. So we expect very good lab results from here. To summarize, so this is a very short presentation of our pipeline. So there are a lot of details that were rushed. And you should be able to use this pipeline on your organism of interest. We're also testing it now in fungi. And the most important thing is to have this detection of your monophyletic target clade. And because the memory resources now are really low, you can actually use these in extremely large data sets at a range that previously was not possible. And this pipeline is composed of these 3 tools you find here. So neighbors for finding your targets and neighbors, FIR for finding your unique regions, and PRIM for designing and testing your primers. And if you're interested in the full thing, we have a full documented pipeline called MapPro. And with that, I would like to thank you for your attention. And if you're interested in one of the other details, feel free to check our posters downstairs. Thank you.
Thank you. So we have time for one question.
How does that great talk, cool tool? I'm going to try that. How would that scale on like several thousand or millions of genomes? Is that something you think is doable? Is it
millions? I never tested. So actually, from this pipeline, the most time-consuming resource is the blast check at NT for primers. So the inferring the phylogeny on the first step is done with a program called Phylonium, which up to 1,000 genome is extremely fast, like matter of seconds. So it scales up extremely well. I've never went up to 1 million, but I think the maximum I got was like maybe less than 2 hours for, I don't know, 10,000, something like that. But it should scale at least 10,000, 1 million, I don't know. Yeah.
We'll see. Cool. Thank you.
Yeah.
Thank you.